Again, we'll record this and post this on our website. I'll give you the directions to that at the end if you miss it, or if any of it you want to recapture. I'm going to actually share my screen so we can get started as folks join. I'm having trouble sharing my screen today. All right, can folks see my screen? And so, beautiful. All right, I'm gonna just quickly sort of intro and MC, and then turn it over to our speakers for today. Welcome to our July installment of the Knowledge Portal Network webinar workshop. And today is a bit of a sneak preview for you, so hopefully it's exciting. I'm really happy to be joined by Sally Fairhan and Mike Knowles, who are two collaborators of ours who are going to introduce the new community portal that we've built alongside working with them for neurodegenerative disease. And so that's exciting today. So we'll hear from them shortly. And today really the focus is a sneak preview of a new resource. We've been very lucky to be funded to build through the NHGRI for common disease. And so I'm going to give a brief sort of context to it before I hand it over to Maria, who's going to give you a tour of its major features and tools and where it is now. And any of you who are familiar with our current portals will see a lot of this before. And really what today's goal is to set the stage for what we hope to be an official launch at ASHG in October this year of this resource for addressing all of common diseases along with community portals therein. And so today is really about sort of to introduce you to the resource, test what's already been built for the beta version, so you can be our testers, and give us some feedback as we continue to build and grow this resource. So I think the context is the best, is that I think the best to give for context here is that I see that our field, having been in this field for 20 years now, is shifting away from discovery toward new challenges. We've done a really great job in the face of discovery, but it's a bit overwhelming, I would say. And many of the papers, 10 years ago were GWAS studies, sequencing studies, focused on a given trait to discover variants associated with a given trait or disease. And so in many cases, you go from here, you get a locus with a set of variants associated with your given trait, but you need to go to here, which is a mechanism, a specific variant with a specific effect in a specific tissue that you can have actionable knowledge about and potentially either target or at least have some therapeutic knowledge. This is the challenge that we're facing. And I think in our field, things have really shifted where you're no longer seeing papers, with just a GWAS. You're no longer seeing papers where they just assay, tissues. You're seeing papers that are more like this sort of really elegant tour de force that just came out from Katten and Sustek's group for kidney disease, where essentially they've run the gamut, where they have, tackled the GWAS, done the credible set analysis, done the ascertainment of perturbations in relevant tissue types with disease states and non, and done the methylation QTLs, linked it to animal models, and pull it all together into one paper that they publish. And this is an elegant paper in my opinion. What's lovely about this, moreover, is she worked very hard with us to make these data accessible. So you're seeing more and more of these in the field. However, there's still a challenge here. There's a gap. Making this information accessible, facilitating the process for those who are actually doing it. Meaning, if you can generate results for computational approaches on GWAS data sets and make those accessible at hand for someone who's studying a trait, generate data and aggregate it for relevant tissue types and genomic annotations, make them accessible in one infrastructure, such that someone doing this work in another disease can have access to it. So it sort of like narrows the time it takes them to do a study like Catlin did. And for folks who don't do this work at all in sort of a pharmaceutical setting or industry or basic biology, facilitate a resource for them to view these data, not just in a paper, but in an interactive resource that attempts to present it to them in a fashion where they're not a disease expert or an expert in a given data type or domain, they can interpret it. That's our goal. So with that, the beta version of the Association to Function Knowledge Portal aims to be a community genomic resource for common disease. And the aims are very simple: aggregate high quality data sets and bioinformatic methods across 7 major disease classes for common disease, integrate methods and data from community experts and publicly available data into one comprehensive resource, and then develop access mechanisms, whether it be programmatic APIs or web-based interfaces to access this information. And our mission to do this is to collaborate and create relationships, some of which we already have, with communities who are actually doing this in the disease type, in the data, in the cell type, or in the space that we need to aggregate the data. Work with them directly to transfer the methods, the data into the platform to develop this common resource and make them all accessible. What's beautiful about this is so many communities come at this from different perspectives. And we also felt that if you created one resource that tries to do this in a static way, that's good because so many of the foundational tools and approaches are the same, but each community has a different lens by which they do this. So to address that, we've built, the common metabolic disease knowledge portal, as you may be familiar with, and we also have done this in many other diseases. But our goal is to address this for common disease and do this more broadly. So we've made some inroads the past couple of years. You've heard about these in webinars. We started with the type 2 diabetes knowledge portal, expanded to cardiovascular disease, cerebrovascular disease, musculoskeletal disease, and on and on and on. But to do this at scale, we have to work with these wonderful communities who have signed up to do this. And the resource will hopefully address these key questions. For our associations that we're seeing in complex traits, what is the variant? What is the regulatory effect and what tissue? What is the gene ideally? And then ideally the mechanism and pathway. So present methods, data, resources, and tools to address these questions holistically. So with that, it's publicly accessible, but it is beta, and Maria is going to take you through it. Beta being, you're going to see it change day to day. It's in the infrastructure of our current portals, but we are going to be overhauling it so that it's much more user-friendly and customizable, where you could potentially view diseases based on genetic similarity or based on disease similarity. So different ways of viewing the data, and also new features and tools will come on board. So Maria's going to talk a little bit about that. But right now, what you can browse is over 412 traits, diseases, across 435 traits. So all of the data sets that are currently accessible on our portal are all brought together, along with some new ones. And we'll be updating that with a formal launch in October. And what I was mentioning earlier, each disease community comes at this differently. So with the broader lens across A2F, you also, association to function across common diseases, you want for each disease community a way for that community to represent their own distillation of their results, any methods that they might have contributed themselves, or presentations of knowledge that are, curated specifically by them. So to that end, we've created community portals, which is essentially the same data, the same infrastructure, but the custom content and presentation for a disease community with its own URL, actually, in many cases, that allows them to have a lens into this resource from their specific perspective. And so we're working with over 30 disease communities to build these resources, some of which you've already seen. But today I'm really pleased that Sally and Mike have worked with us, along with others in the neurodegenerative disease space, to bring together a resource for neurodegenerative disease. So we're going to talk a little bit about that in a minute. But first, a little tour from Maria, and then we'll take questions at the end. Feel free to pop them in the chat or in the Q&A, and we'll address them if we can along the way, or we'll address them at the end. So with that, I don't see any questions, so I'll turn it over to Maria.
Okay, let me share. Okay, well, I'm not going to give very much of a tour because if I'm suspecting that most people on this call are familiar with the Common Metabolic Diseases Knowledge Portal, and right now A2FKP is just the same, or very much the same. So we have a similar homepage, and again, we can enter by gene, region, or variant, or by phenotypes. And if we type a gene name into the box, I'll use our standard diabetes example, we come to a region page. And the difference here is that now we're looking at more traits. So you may see other phenotypes that are not in the CMDKP. So this, as you know, the region page shows significant associations across this region. We also have the Genomic region minor module where you can stack up locus zoom plots for different phenotypes and look at epigenomic annotations. The variant sifter, our new tool for prioritizing genes and variants, along with epigenomic information, is also accessible from the A2FKP. And it can draw on all of these traits that are in the A2FKP. And basically, we also have gene pages with all the same tools and features so far. So yeah, I'm not going to give a detailed tour of this portal. But what I do want to show you is some of the things we're thinking about that will be different, new and different in the A2FKP. So as you heard, and as you can see, the number of data sets and traits are really on a different scale in this portal than any of our other portals. CMDKP is our biggest current portal. And A2F is only a little bit bigger now while it's under development, but we're expecting to add, maybe 100 to 200 new data sets a year to this. So it will quickly become huge and possibly unmanageable. And even now, I know you can't really see this, but this is a screenshot of the page that lists genetic association data sets in A2FKP. So it's pretty long. It's, basically unnavigable at this point, even with the number of data sets we have now. And we do have a filtering mechanism, so you can filter these data sets by their phenotype groups and their phenotypes. But even so, it's kind of difficult to navigate. So we definitely need to address this issue in A2F. So I want to talk to you about 3 lenses for viewing the A2FKP and getting into the data. And one of them is by physiological system. So to let people navigate the portal and choose the phenotypes that they're interested in, we're going to design a relatively simple ontology. And this will have physiological system, such as cardiovascular system, as the parent. Under that will be the diseases of that organ system. Under diseases will be phenotype groups, which we already have in the portals. For example, lipids is plasma lipids, and cardiovascular traits are things like hypertension, heart rate, and so forth. So each disease will be apparent to the groups that are relevant to it. So for example, here we see coronary artery disease. All of these 3 groups are relevant, but for atrial fibrillation, maybe plasma lipids are not so relevant. I mean, choosing these is going to be done in consultation with the experts in the field. Then each phenotype group, as currently, has a bunch of phenotypes within that group. But a change that we will make is that we're going to make it so that one phenotype can belong to more than one group. This is really important for accuracy, I think. So, for example, currently we have phenotypes like diabetic kidney disease. Well, that should be in the diabetic complications group, but it should also be in the renal group. So, and it's not currently; it's only in one. So we'll make that navigable. So we're going to construct this ontology, and we are aware that there are other ontologies out there for phenotypes and traits. There's Mondo and EFO. And we do want to map each of these traits to those ontologies so that we can be interoperable with other resources like the GWAS catalog. But we don't just want to implement those large ontologies in the A2FKP. We feel they're too big, and it would be actually more complex than is necessary. We think we can do this with a relatively simple and small ontology at this point anyway. So we're constructing this ontology, and then this will allow users to browse by physiological system. So you might click on the cardiovascular icon, and then you'll see the diseases. You might choose coronary artery disease, and then you might choose a couple of the groups that are under that, and so forth. We're also going to allow users to browse the phenotypes by how closely they're related in the ontology. So for example, hypertension is going to be closer to HDL than, say, to fasting glucose. So we'll let people browse by that criterion as well. And then we are also going to enable them to create and save custom phenotype groups. So, if I'm interested in ECG traits and MRI traits, I can put them together into my own custom group and look at the data for those traits. So this is a really, manually curated approach. But then we want to offer a complementary approach. So we also want to give people a lens to enter the A2FKP by a data-driven approach, and that is genetic similarity of phenotypes. So there's a new paper in Nature Genetics by a bunch of very prominent scientists, and it deals with a method for figuring out genetic covariance, that is the correlation across the genome for variant effect sizes of any two traits. And all you need is GWAS summary statistics to run this analysis. Here's a figure from their paper just to illustrate it. So for example, so blue is positive correlation. We see here that BMI is positively correlated with T2D, and HDL is negatively correlated with triglycerides. So, I mean, we knew those things from medicine, but the genetics support it as well. And this kind of analysis may bring up connections that are not necessarily so obvious. For example, type 1 diabetes has many symptom similarities to type 2 diabetes, but its genetic architecture is actually closer to autoimmune diseases. So, connections like that may come up from this analysis. So we want to offer people a way to navigate this. This is just a wireframe that our UI designer DK made to do this. So the user would select a primary phenotype and then would get a list of the phenotypes that have a certain degree of similarity, above a certain threshold to that phenotype of interest. Then they could select a custom list of those phenotypes and create their own custom group. Farther in the future, we want to also use these data and cluster traits by their similarity, and then allow users to select the clusters that they're interested in. We're still working on implementing this. So a third lens, of course, for viewing the A2FKP is the community portals. And you're going to hear in a minute from Sally about the newest one, the Neurodegenerative Disease Knowledge Portal. But before we go there, I want to talk about one other area in which we're really investing in A2FKP, and that is effector gene predictions. So just to clarify what I mean, genetic studies of common diseases usually come up with a few hundred loci in which there are significant genetic associations for that disease or trait. And in those loci, you can do fine mapping to identify a causal variant. But then unless that variant, say, changes the coding sequence of a protein and creates a loss of function mutation, in that case, it's kind of obvious what's probably happening. But in most cases, that is not the case. The change, it's not obvious what the change is doing. And there are a bunch of genes in the region, and we need to identify which one is impacted by that genetic variation, which is the causal gene or the effector gene. So a lot of communities are starting to make these predictions to prioritize the genes that are in the area of GWAS signals. And they're integrating lots of different data types to do this, such as genetic epigenomic annotations, such as open chromatin, gene expression, and EQTLs, model organism evidence. And so they're creating lists of the genes that are the current best guess of this research community, that are the ones that are likely to cause the disease. So lists like this can help researchers prioritize genes for further study, and they can also identify genes that are unstudied or understudied, but still meet all the criteria for being a likely effector gene. And really importantly, these lists allow non-geneticists to access this information. They're not going to go and analyze the associations or the epigenomic annotations themselves, but they can look at the list and see, what does the community think are the most important genes here? And so we are starting to collect these lists from the community, and here's one example. We present them in these interactive tables. So this is a list of osteoarthritis predicted effectors. And the top view, the default view of the table, just shows the summary. So here there's one gene per row, and then there's a summary there's a scoring system for each list, and summary of some of the top information. And this may be enough for the non-geneticists just to see which genes are on the list and what their scores are. But then if people want to explore the evidence, you can click the view button, expand some rows of the table, and see the detailed evidence. And often these lists have links back to the provenance of those pieces of evidence. So For A2FKP, first of all, we want to collect all available lists. And we've been doing a systematic review of the literature to find them. And it turns out there are quite a few for many of the major diseases and traits. And so we work with authors to get these lists and their supporting evidence and implement them. There are also automated methods for making these predictions, notably the effector index method from Brent Richards' group. And we want to implement methods like this in the portal so we can do them at scale on all of the traits that are in the A2FKP. And then once we have all this information, of course, we need to make it available throughout the portal. So right now, our lists are collected on one page, and that's fine for now when we only have a few lists. But as we get more, obviously, we need to make a better interface for browsing and searching this list to see this list of lists to see what we have for which traits we have effector lists. But we also need to make this information available in a few more places where it's not currently available in any of our portals. So for people entering by a disease or a trait, when you're interested in that disease or trait, you would want to see, is there an effector list for this trait? Or are there multiple lists, in fact? So we definitely want to make that information available via phenotype pages. And I think most importantly, actually, we need to make this information available per gene. It's not at all currently, but if you go, say, to a gene page in the portal, which tells you everything known about that gene, that page should definitely tell you, okay, this gene is predicted to be an effector for X, Y, and Z traits, and lead you to be able to see all the supporting evidence. That's key. So we are definitely going to do that. Okay, and I think with that, I will hand it over to Sally, who will go back to the third lens for viewing the A2FKP.
Perfect. Thanks very much, Maria, Noelle, and Mackenzie, and Mike for all being here, as well as all of you. So I just wanted to give a brief background to the origin of the Neurodegenerative Disease Knowledge Portal, NDKP. And first, I just want to thank Jason Flanick, Noelle, Maria again, and Mackenzie for their leadership, their collegiality and collaboration. It's been a pleasure working with them, and I look forward to continue working together. So really, the origin of the NDKP began with an ALS-KP, so the Amyotrophic Lateral Sclerosis Knowledge Portal. And the goal for that was to display the results that we had published from a 2019 paper so that other people can access this large data set, the summary statistics of it, as well as other genome-wide association studies that were out there as well, focusing on ALS. In addition to ALS, we have wonderful colleagues like Mike Nals here who have been doing tremendous work on other allied neurological disorders. I call them allied because there are times actually where patients can present with ALS as well as cognitive impairment. That cognitive impairment can go on to actually be a distinct clinical phenotype called frontotemporal dementia. Additionally, some patients can present with Parkinson's-like features, as well as other overlapping neurodegenerative diseases. Now, these are distinct clinical diseases. So if there are clinicians on the call, they would say, well, I would argue that they are distinct, and I completely agree with you. And the genetic architecture can be distinct as well, although there is shared genetic architecture. And is that because of what we know, or is there more to be discovered? So, I thank my colleague, Mike Nels, for joining me in this effort. And basically the goal was to really advance other neurodegenerative disease studies to what he and his team at the NIH has done for Parkinson's disease, what they want to do for Alzheimer's disease, among other allied neurological disorders as well. So that really was the goal, is that for other members of the neurodegenerative disease communities to know one another, get to know our data more intimately, and also have one consolidated resource where we all have a place to put all our data as well. Additionally, too, the GWAS catalog, as Noelle and Maria alluded to as well, has been an excellent and well-reputable resource. But it has everything, right? So it would be nice for commercial places, academic places that want to specifically study and target neurodegeneration, they have one consolidated portal to use. So if they are interested in a target that perhaps is involved in a pathway that's involved in neurodegeneration as a whole, this would be a good avenue to explore. Or they just want to just look at ALS, for example, they could use that portal as well. So we've been going back and forth with the type 2 diabetes team to try to leverage what they've already done successfully so that we could advance our field and take advantage of those resources as well. So I think Maria is going to give you a demo of what the Neurodegenerative Disease Portal looks like. It is also in development as well, although they've done a fantastic job. And for anybody in this audience here as well, please give us feedback. We would love to hear more from you and your colleagues in ways that we had not thought about it, or ways that we could implement additional features as well, and then also additional data sets as well. Our goal is to encompass other neurodegenerative disease phenotypes, and as they become publicly available, or as scientists want a place to deposit their resources as well, we would be facilitating that as well. Mike, did you want to share any other comments, or does that kind of summarize what our goals are so far?
Yeah, that was a really good summary. I think one thing that's really nice about this, as a fundamentally lazy person, not having to click too many times to find data on related dementias or Parkinson's or ALS, and have them in a directly comparable space is really useful. And it's also great because just working with this team has been really rewarding because of the flexibility and openness to kind of boutique GWAS, which is something you don't see in a lot of places. So thanks for all the help.
Thanks very much. So Maria, I'll hand it to you to give a demo, and I'd be happy to also answer any questions from anyone here.
Okay, great. All right, so here it is with a pretty new icon, again, from DK, our designer. The structure of the NDKP is quite similar to the other portals. So if you're familiar with them, you'll have no trouble navigating it. Let me start by showing you the data content for genetic association data sets. So these are the phenotype groups that Sally and Mike have decided are relevant to neurodegenerative diseases that they would like to group into this portal. And those are flexible as we change groups and add groups, that can change. But the goal is just to amass all the disease relevant phenotypes. So if we just look at neurological diseases, we have a variety, Alzheimer's, LBD, ALS, and we're looking to add a lot, and Parkinson's, of course, and we're looking to add a lot more, particularly in Parkinson's. And so those are the data sets, and I'll just give an example of navigating through. Well, first I should say, though, that this is a community portal, and so it has some, and it will have more community-specific information. So here, the About page describes, right now, we're just describing the ALS research effort, the consortia that generated the data and so forth. And we'll be adding more information to this page to describe the other disease efforts. And so let's go back home and let's just look at the SOD1 gene as an example. So like the other portals, when you enter a gene on the home page, you get to a region page that shows the gene and the region of the gene. And so here, ALS is definitely the strongest association. There are some others. These associations here are just all of the most significant associations across the region, but this is the only genome-wide significant association here. And these are clumped variants. They're clumped by their linkage disequilibrium. And you can expand these rows to see all of the individual variants that went into the clump. Well, that's interesting. This is a clump of one. Some clumps have many more variants. Here we have the Genomic Region Miner module, which will let you see a locus-zoom plot of the associations in the region. You can stack up plots for multiple phenotypes if you like. And you can also add tissues and annotations, epigenomic annotations here. So here we've added these blocks represent regions in that are annotated as being either promoters, enhancers, and so forth in the central nervous system tissues. So here, central nervous system is a sort of a grouping term, and there are more detailed tissue and cell types within that group. So you can see, for example, if your variant of interest lies within any epigenomically annotated regions of interest. Here it looks like it's within an enhancer in central nervous system. That could be significant. As many of you know, I'm sure we're also working on a new module, the variant sifter, that does the same sort of thing, but is more versatile for prioritizing genes and variants. And so this is also available in the NDKP. And we can filter by annotations here. So let's look at enhancers, since that seemed to be significant. We'll look at central nervous system enhancers. And so here you see a plot showing the locations of enhancers in this region in central nervous system tissue. These are other tissues. And we can look at our index variant, mark it with a star. And now down here, this vertical line shows where it is. So it shows that it's in an enhancer, this enhancer. And it looks like it's also in enhancer regions within some other tissues. We can also filter by linked genes. Let's look at the central nervous system. So what this is showing us is regions that are linked by target gene prediction methods, such as the ABC method developed in Jesse Engritz's group. So these regions are linked to specific genes. And for example, if we look at our SOD1 gene here, so this region is linked to the promoter of the SOD1 gene in bipolar neurons. So this is another way of exploring these variant gene connections. In this case, our variant of interest, our index variant, isn't in one of these regions, but other variants would be in other regions. As you make all the selections in the variant sifter, the table here gets filtered down to only list the variants that are answering all of your criteria, or the intersection of your criteria. So you can really choose your criteria and get a prioritized list of variants that are of interest to investigate further. Like the other portals, this portal has gene pages. So I'll go to the SOD1 gene page here. This is meant to just collect all the gene-specific information. So we've got information about the gene and its product from external sources here, just to orient you. We have the huge calculator, which allows you to look at the genetic support for the involvement of a gene of interest in a phenotype of interest. Let's just for fun, okay, well, it's not telling us that SOD1 is involved in Alzheimer's. Let's see. Compelling evidence that this gene is involved in ALS. What this tool does, and you can get more information about what it does on the tool page itself, but it looks at both common variation and rare variation across the region, and does a calculation to come up with a score. Here's a PheWAS plot of gene level associations calculated from common variant associations using the MAGMA method. So these show these are just different, all the different phenotypes across this portal. This gives you the MAGMA values for this gene, for those phenotypes, and they're listed in the table here as well. Rare variant, okay, this feature, rare variant gene level associations, is draws from a restricted list of phenotypes that were done in one study, and those phenotypes are not applicable in this region of this gene, so that we're not seeing that. Okay, I'll just show one other type of page here, the phenotype page. This page collects all the information about associations of a phenotype across the genome. So this plot shows our bottom line meta-analyzed genetic associations. We take all of the data sets with associations for one phenotype and do a meta-analysis using the METAL method developed by our colleagues at University of Michigan. It does an overlap aware analysis, so it accounts for sample overlap between data sets. And we do that meta-analysis and do some other steps. So we're actually calling this integrative analysis to come up with sort of the best guess or gold standard set of annotation of associations across the genome. And you can see the top variants that came out of that analysis. You can see the top genes that are associated with this phenotype. And finally, you can see which data sets went into that meta-analysis. Each data set is also described in more detail on its data set page. You can see the associations just in that one data set, both the plot and the table of variants, and then there's a description of the data set. I think that is basically the tour of this, this portal.
Can I just ask a question, Maria?
Yes.
I recall you're able to include multiple phenotypes. For example, could we do where we look in one gene, but we look for ALS as well as Parkinson's, for example, just to see if there is one, like
Sure.
one gene that's a big influence in ALS. What does it look like in Parkinson's or Alzheimer's or something?
Yeah. So the SOD1 gene didn't have any significant associations except for ALS. Do you know of a gene that would have?
Yeah, let's try MAPT. M as in mom.
MAPT.
As in Maria. APT. Yeah, exactly.
Interesting. That's not coming up.
It's not coming out. Okay, maybe is
Oh,
it?
in a minute. No, it is.
Yeah. Okay. Perfect.
While Marie's doing that, we also have some questions in the chat that we'll turn to next.
Okay, great.
Good to answer across the audience.
Wow, this is an interesting plot. Okay, these are NAPS associations. That's I wonder if there's something funny about these data, but anyway, we can use this to illustrate this. So okay, let's. You want to do Parkinson's?
Sure, we could do Parkinson's, and you could add multiple or just one at a time.
Yeah,
Oh,
you,
okay.
you can add multiple.
Okay. Yeah.
Huh.
Do Alzheimer's. It's definitely a. Or yeah, we can do ALS.
Yeah, you can add as many as you want.
Yeah.
Wow. I have never seen a locus-zoom plot like this. Maybe someone watching knows how to interpret this line of variance.
Yes, and there's a big LD block. I don't know. That's interesting.
Oh, just a huge
Yeah,
LD block.
I
Could
put it
be.
in the chat. It's just a big, very old LD block.
Oh, interesting. Okay.
Old as in human evolution old.
Somehow
Okay.
nature's preserved that.
Wow. So yes, we can stack these up. We can use the crosshair to see where the most significant Alzheimer's variant lies in relation to the ALS variant and so forth. It looks like all of these diseases, the variants are fairly close to each other, even NAPS. Okay, did we have more questions?
Yeah, we have one that I think potentially, Mike, you might be best positioned. This is a question from Doug Keel. How does this new portal interface with similar NIA efforts like the AD knowledge portal.synapse.org? Mike, do you want to field that, or I can try as well?
Yeah, I mean, I think the Synapse portal is really a repository for the AMP-AD project, and it's not really such a focused kind of GWAS browser, right? I mean, there are a lot of fractured efforts across different kind of data silos, right? You've got, 15 different groups doing Alzheimer's GWAS all within a month of each other, when the UK Biobank released data. So it's nice to have the ability to push a lot of different data into one place from disparate sources, right? And it's actually, in my opinion, quite nice. It's a third party-ish, so to speak. It's not quite third party, but it's you're working on so much, particularly Noel and Maria, that everything is there just for the community resources.
Yeah. I think to like riff on that, I think one of the things we're hoping actually is, by virtue, actually Mike can comment on this as well, in doing this effort, we were very lucky. Now we're sort of creating inroads with a consortium that Mike is involved in leading for called GP2 for Parkinson's. They are generating data, have their own resource to make the data accessible for the data they're generating, but creating a partnership with them creates sort of reciprocal data sharing or reciprocal cross-linking, where they have at least either represented the results in a better way, or they're more advanced, or they want to share their API with us. So that's the goal, is to create these connections with these conversations. So AMP-AD, we're funded by another AMP initiative, and so our goal is, now that we sort of have this, we can actually talk to them and say, where might we actually create connections and synergies and ways to view your data in the context of ours, and vice versa, so that it is more harmonious, but allows it to also develop in their own, respective mandates. Sorry, Mike, did I cut you off? I apologize.
No, I was just gonna co-sign kind of what you're saying, is that, but with one kind of thing that maybe some people miss this, because they're kind of drinking from the fire hose of information right now, because you presented a lot today, is that things like AMP-AD are really good, and there's a lot of other resources that have multimodal metadata that can then later be incorporated into what the team has presented today, so that you can really leverage summary stats, not just from GWAS, to do this and prioritize variants and genes. It's really flexible, right? So like AMP-AD isn't really just a GWAS repository. It's got a bunch of cool metadata and things like that in it. Whereas other sites, other sites are focused on GWAS, on sequence-based association studies, and you can kind of bring everybody into one place, which is great.
That's what you're seeing.
It all goes back to me just being foundationally lazy and not liking to click things.
I think what you're seeing here is a lot of this is like you have to request access to it, because this gets you, I think, in many cases, actually the real data. And so that's not the goal with the with the A2F or the DKP. It's sort of an integrated representation of the summary representations of the results of those data. And our goal actually is, where there's individual level data or, and complete metadata for example, epigenomics, proteomics, as you see here, we would link out to that, so that if the sort of the distillation of those results are presented in the Neurodegenerative portal, you could actually get to the raw results if you were so inclined to get them. So that's what's the complementary approach of many of these resources that we're trying to achieve here. So a couple other questions from Andrew Patterson. I think this is more about actual data, which these are some good feedback. One is the region plots look very similar to my locusm plots. They are by the fact that, we work with Michigan actually to build this resource. But one of the limitations he's noting is that it's not possible to zoom in and zoom out. So Maria, do you want to answer that and why we've done that and what we're thinking there?
Yeah, could you stop sharing screen and I'll share my screen?
Of course. Good point. Sorry. Oh.
Okay, so Andrew, were you referring to this plot? Let's see, can we let Andrew speak? So you can answer that.
Yes, I think I can. Hold on one second. If he wants to speak, that is. We'll put you on the spot, Andrew.
That's right.
So were you referring to this one?
Yes.
So it actually is possible to zoom. If you hold shift and scroll.
Oh, I see.
And zoom in and out. So that is possible. And so you also said you would like to see the, more information in these tooltips. So yes, I agree. That's great. And we should do that. For the time being, you can see some of this information. If you scroll down in this table, this shows the significant variance in this interface. So for example, here's that index variant, and you can see the p-value and the odds ratio. And an alternative view of this is in that variant sifter that I showed. And here you can also zoom in and out using these controls. And in the plot below, you do also see this same information. So, it's not in these tooltips. I agree. It should all be in the tooltips. And something I've been advocating for is to see the RSID in the tooltips, because the RSID is what everyone uses in papers, and it makes it much easier to correlate what you're reading in the paper with these, if you can see that. But So yes, thank you. I think we will add those things to the tooltips.
And that table that you showed just at the bottom of this also had the allele frequency, which I think is also very important.
Yes, so it has a minor allele frequency.
Just one other comment. When you first started and you showed the studies with the number of subjects study, to me, what would also be very helpful would be to break that down by the number of cases and controls.
Ah, yes. Okay. So here, each data set page will show the number of cases and controls, I believe. Yeah. So, but it's just kind of in free text here. You'd like to see that right in the table.
Yeah, because let's say there were 10 Alzheimer's disease GWAS results, and I wanted to choose what I thought was the most powerful of them. Kind of having that information would be the most obvious way for me to select.
Yeah, this makes sense. Thank you.
No, that's a really good point, Andrew. I think that's something that, particularly for disease data sets where we have like hundreds of data sets now, right? You want to direct the user to the one they should use the most, or give them the information that they need, including more detailed information about ancestry as well. Because you'll notice in the portal, right, we collapse everything into a bottom line meta-analysis that accounts for sample overlap and presents you sort of like a final p-value, where many cases you as a user may want to know, how many GWAS went into that, how many samples, what's the effective sample size, what's the ancestry. So that's captured in our bioindex and in sort of our backend. And we're going to, in our next sort of iteration of the portal, we're going to expose a lot of those statistics. So you can see like, what's the effective sample size for the full meta-analysis we did? How many cases and controls went into it? What are the different studies? We have that right now, but we don't have the information that sort of subsumes that into like a summary. So that's really good feedback. And something we need to make sure we do with our next production release. Other questions? It's nice to see some new faces on the webinar. I think one of the things that, the reason we did this today is, frankly, we have some awesome people to work with across the common disease space to give us feedback, but we're always looking for more, particularly someone who may have data they want to contribute. That's always great. But really, actually, one of the places we made a lot of progress, and sort of Mike's been really helpful in this example, is, thinking about different, when you're working on a specific disease or trait, you have a specific approach where you might have a method or a pipeline that you've instantiated for your particular disease. What's nice about the software resource we built is it's relatively straightforward for us to implement or encode a new approach or a new method and make those results accessible. So one of the things that just, if any of you are methods folks, just to sort of put a plug for that, or running bioinformatic predictions or creating curated lists of effector genes for your given trait, we would also like to see this resource as a repository for folks who are doing those approaches to share their results, but also potentially enable those to be run on other traits and made accessible. We've done this with several effector predictions across 12 traits with Brent Richards. We've done this with Melina Klausnitzer to run her Bassett method across all of the traits in the portal. So these are things we're beginning to do more of. All the gold standard ones we're implementing, right? So right now you'll see LD score regression will be coming online this fall across all of the portal traits for tissue enrichment. So that's really, that creates the wealth of the rich resource. So not only do we want to get more data, but we want to get better methods to prioritize genes and variants. And people involved who are just interested in bashing at the site and tell us what works and doesn't. Any other questions? All right, a couple closing announcements. Let me share my screen real quick. I want to thank Sally. Particularly, Sally came to us, oh gosh, 2 years ago, and she mentioned it. She's like, I want to build an ALS portal. It was literally, a one-woman show, and so she got it done, and that created the beginnings of some beautiful relationships that led to Mike and others collaborating with us in Neurodegenerative. So I want to thank Sally for that. That's been really great. So in August, we have our next installation of the webinar series, and it's going to be a scientific session. One of the things that's really nice about some of the funded efforts that build the Common Metabolic Disease Knowledge Portal is not only are we generating, building the resource, but we're also generating tissue-specific data in different cell types and tissues and perturbational screens for common metabolic disease. So we're going to hear about the research going on within this consortium next time from Klaus Kessner. He's going to talk about his recent paper for three-dimensional chromatin structure and the pancreas cells for the architecture of type 2 diabetes. So he works with the HPAP consortium. Again, this is another consortium that we're connecting with to build the resource and enrich it with data. But he's going to talk about his approach. Ultimately, these data will be accessible in the portal, but it will be really a scientific session hearing about the research going on in the common metabolic disease for AMP. So with that, any other questions? I don't see any. I really want to thank everybody for joining. If you aren't on our mailing list or know people that we should be reaching out to, please shoot me a chat or go to kp4cd.org. That's where actually I'll just show it to you so you know what it looks like if you land there. It's right here. This is actually the resource for everything soups and nuts, from the platform that we build to our other platform where you can actually build your own portals if you're interested and publish those. All the knowledge portals in the network, all the resource portals we've built for your build your own portals, and then all of the news. So you'll find today's webinar and everything else from our tutorials to our how-to guides to videos for different scientific presentations accessible there for your viewing pleasure. So thanks for attending. Thanks to our speakers, and hopefully we'll see you in August. If not, happy summer. Take care, everyone.
Thank you so much.
Thank you.
Thanks. Bye-bye.
Bye-bye.